Okay, well, good evening. Nice to be here. When I accepted this talk a year ago or something like that, I thought I would start by explaining what a virus is and what a pandemic is, but I guess we don't need that anymore. So the story changed a little bit. So my overview is really starting at this point. What happened? I will do a bit of a look back at the current outbreak from a scientific perspective, and then go into a bit of the context behind it. And that's the type of research that I've been working in for many years now, preparedness research. That's the heading that I enjoy. So this is where the current outbreak started for me. This is a message that was sent around on Twitter from a Chinese financial newspaper, December 31, and they had picked up a leaflet from the Municipal Health Service in Wuhan, in the province of Hubei, where they warned about some type of pneumonia, that there were people with pneumonia and it was related to a market, and they also said they were doing a very good job at finding out what it was in a bit China style. So that was the first alert about this virus, which was also then shared with the World Health Organization. Now, my first thought was, oh, could this be an avian influenza virus? Because that's the kind of viruses that we know are quite common in wild birds and in domestic birds in the market system in China. We have some studies ongoing, and if you do environmental sampling on the live animal markets in China, 10–20% of samples will test positive for avian influenza viruses. And that can be a problem, because we know that these viruses every now and then can spill over to humans and then cause disease, and in some instances cause severe disease that becomes transmissible, pandemics. And influenza is, of course, well known for that, notorious for that, but also well known for that. We've seen over the past 100 years or so this sequel of pandemics, the most notorious one, the Spanish flu pandemic, 1918, then Asian flu, the Hong Kong flu, Russian flu, and you may remember this, 2009, the swine flu, Mexican flu. You cannot say Mexican because that's, well, Mexicans didn't like it. You cannot say swine flu because the swine farmers didn't like it, but that flu anyway, 2009. And then, in addition to that, there's a constant trickle of zoonotic cases, people that get sick with a virus, influenza viruses from animals, but in this case, just a trickle of little incidents, but no spreading viruses. That's the situation that we know for influenza. But no, this was not influenza. The Wuhan Municipal Health Service mentioned that very fast. They tested for that. But then this was circulated again over Chinese Twitter. It was retracted very soon after that, but that said this was a lab report, and it mentioned the word SARS, RNA. So that triggered a lot of attention. Hey, what's going on there? Apparently, people had found something that might look like SARS, and SARS is a virus that we know because it had been causing a big outbreak. It's coronavirus, a big outbreak of severe respiratory disease in around 2003. And yes, that turned out to be correct. So a few days later, the formal release of that information came first through a global data sharing system for genetic data of viruses. But soon after that, also a full description here in this publication, where they showed cells, infected cells, with viruses in them. So this is for a person like me, for a virologist, a very convincing picture. These here are viruses. They showed the genetic sequence of the virus, the genetic code, and they showed where that fits in the whole tree of coronaviruses. And they showed that people with this infection were developing, after a little while, were developing antibodies. And that combination of experiments and studies really is a very solid way of demonstrating there is a new virus and it is the cause of this disease. It's really associated with this disease in a very short time span. So I'm one of the people that thinks this was pretty impressive. Now, with that sharing and with that release, of course, we learned a lot right away. We learned immediately, where does this virus sit? So, as said, a coronavirus. Now, coronaviruses is a very diverse family of viruses. Here they are. They branch in many different categories. And as you can see, there are coronaviruses for many different species, including humans. Here are some of the coronaviruses that we already knew. Here and here, coronaviruses that are part of our winter cold package. 4 coronaviruses that have circulated for a long time. You get them, everyone here has had one or more of these infections in their lifetime. But here then, the new coronavirus fitting nicely together with SARS coronavirus, so closely related, fairly closely related, and matching with a virus, SARS-like bat coronavirus that had been found in bats. So just that genetic information already tells you quite a bit. So not one of the known human coronaviruses, a virus that is eerily close to SARS, which we knew had severe impact on humans. So now we're looking at a spillover of an infection, a virus infection that probably has been circulating in animals for a long time, but trickles over the fence or out of the tree, whatever you would like to think, into humans. And then these are zoonotic infections. And the moment you see a zoonotic infection, an immediate series of questions is, what does it take to understand whether this is a one-off event, just a spillover that dead ends because a person gets sick, maybe dies, think of rabies, for instance, and then that's the end of it? Or is this a virus that is transmissible among people? And if so, is it transmissible in a very efficient way? Because if that happens, this can become a widespread epidemic. So with every spillover event, whether that's avian flu, this virus, any virus, there's this series of questions that you immediately have. What's the reservoir? How widespread is it? How contagious is it? And so on. And I will walk you through some of the studies that were needed in order to get at these questions. Let's start at the start. So where did this come from? Well, at present, we still don't know that for sure. What we do know is that the new virus, here it's still 2019 new coronavirus, the first hit was with a bat coronavirus that had been detected in this type of bat species, which is quite common in a wide region of China, including the Wuhan region. And we know that these viruses are in these bats because they have been part of a survey program, a virome search program that was done by a group in the US. So that link was there, but the viruses of the bats and this virus still are quite distant, and there must be something in between. And one of the questions is, what happened exactly with that original seeding event? Has there been a bat introduced on a market or in someone's home for food that then triggered this initial spillover from where the virus spread between people? or has there been an intermediary species, something that we often see, where the virus spills over in some other animal that is more close to humans, and that, for instance, is produced for food. Now, one of the candidates there was this guy, pangolin, that happened to be a popular food animal. It happens to be also endangered and critically endangered, so that's a weird combination. And because of that, these animals are, there's a massive market pulling these animals from across the region, from all over Indonesia, but even from Africa through Vietnam into China just for food. And that's not a Chinese food preference, it's also a very popular tourist attraction. So there's a bit of a system there that really is a dangerous system if you want to introduce strange viruses from a wide geographic region. So in pangolin, a virus was found that is quite close to this new coronavirus in humans, but still too distant to say that this is a direct hit. So that is still a question. What has been in between? Is that source still out there? And that's an urgent and frequently repeated question for the Chinese to answer. Since then, we've learned that there is this virus is fairly promiscuous, and we've seen either natural or experimental infection in a range of animals. Cats, a tiger in a zoo in the US, ferrets, mink, dogs, hamsters, rabbits, bats, of course, macaques, and probably that list will turn out to be longer when we learn more about this virus. Now, really, this is a very busy picture, but it is a picture coming from a paper from the Food and Agricultural Organization that tries to unravel where do we need to look, what do we need to study, what do we need to understand to really ask this, to understand this question. Where is the risk and how are these viruses transmitted? So what they do is a very systematic process where they look at all pieces of evidence that's out there, and they look at what are risks if you think about animal introduction. It could be through hunting, could be through farming, just through the slaughtering process, or through environmental contamination. And then they have a way of looking at pieces of evidence for one or the other. And right now, their conclusion is that it's still, it needs to be investigated, particularly in China. Now, so here we are. This is how we were looking at it in January. A virus that was obviously happily spreading in a family of viruses where we know we have zoonotic viruses that ended with SARS and with MERS that do not spread so easily, but also several coronaviruses that have originated as animal viruses. Not in our lifetime, so this is evidence that comes from evolutionary study, but the evidence is pretty robust that all of the now common cold coronaviruses have had their origin in animal viruses, so these have been pandemics. So that's why the expectation at some point was, well, then why not this virus a pandemic? And that's eventually, of course, what is happening. Now then to the next question, and that's a question, of course, that has been heavily debated, and you have believers and non-believers still till this day. How severe is this, really, and how widespread is this? And I guess if you followed the news and the discussions a little bit, you now know how difficult it is to really answer this question solidly. And why is that? Well, because if you think about infection and disease, you have to think about really an iceberg where what you see is the little piece of the iceberg that sits above the surface of the water, and the rest underneath, you don't know how big that is, because, well, you don't see it if people are not ill, for instance. And then picking up a disease, certainly if it's a new disease, is quite difficult if the symptoms are so common. If it's fever, runny nose, who doesn't have fever, runny nose? So very difficult to identify exactly where is this new infection, who has had it, and who didn't have it. So there's been a lot of discussion about this, but this is typical for every emerging disease outbreak, that it takes months, if not longer, to understand what the full extent of spread is and what the severity is. And the same applies for how exactly does this spread. So this is a famous picture that that is used to teach influenza transmission, influenza epidemiology, and that's what happens if you sneeze. And by now, everyone is a little bit paranoid about this, I think. But yes, sneezing, coughing, even speaking, singing, we've learned, does produce these droplets of different sizes. Many of them are big, and of course the biggest droplets also contain the most virus if you're infected. Some of them may be tiny, and the tiny droplets may stay in the air for a longer period of time. But for this virus, the current consensus, at least in the advisory organizations, is that droplets really are the most important for spread. And of course, spread then happens in circumstances where many people are close together and where there's no, in this case, no ventilation. We know for this virus that there's also shedding in stool, particularly maybe in children, but whether that's relevant for spreading is still unclear. And the same applies, so you do find virus in, for instance, right now we're looking in slaughterhouses, you do find virus on the surfaces, but is that really a way of getting the infection? If you touch a surface and maybe touch your face or your eyes, not sure, but that's where the hand washing guideline comes from. So one of the key questions indeed is then, how do you measure, how do you determine transmissibility? And this is one of the questions where we use models, animal models, because in order to really understand this, you need a bit of a standardized system. So this is a series of studies that was done looking at replication of the virus in this model in macaques, where the same studies have been done for SARS and for MERS. And just like SARS and MERS, with this virus we see pneumonia, we see severe lesions in the lungs, we see inflammation in the lungs, but very different from SARS and MERS, this infection is found in the nose, upper airways of infected animals. That's not what you see for SARS and MERS, where the virus sits deep in the lung. And that's one of the reasons why this virus is so much more transmissible than SARS was. What we also saw is that there's more virus in older animals than in young animals. That sort of sounds similar if we look at people. And what we've seen is that if you then do experiments where you look at animals in contact with each other, so one animal gets the virus, you put another animal in the same cage, and that's a contact transmission, and then you put another animal in a different cage, but with a raster in between so that droplets can pass through, and all of them got infected. So there's both contact transmission and droplet transmission. That's what these experiments show. And this is behavior just like we know for influenza in the same system, and much more so than, for instance, for MERS in the same system. That's the type of experimental settings that are used to quantify this. So here we are. So we have a virus that then is characterized by two important properties. One is how easy does it spread, that's going up, and how ill do people get. And the true impact, of course, is determined by both. So you may have a very happy spreader, but with very little disease. Think of the common cold virus, rhinovirus, which typically is in that category. Then even a pandemic of a new virus, but that with those properties, we might want to say, okay, well, it's just not worth all the effort if the disease is so mild. You can have the other way around, very severe illness, but very little spread. That is, of course, then a problem for the people that are hit, but not for the rest of the population. But here we see a combination, spreading and quite severe disease, and that's why the impact of this virus is considered quite high, and it's ranking here in the same corner as the 1918 Spanish flu outbreak because of the properties that are seen so far. Now, and that's the type of pandemics that public health has been preparing for. Flu pandemics. This is a simulation that has been developed by Bill and Melinda Gates Foundation just to draw attention to what impact could be of a pandemic. And you can look it up. It's a movie that shows if you had a new influenza pandemic that infects 30% of the population with a case fatality rate of 0.5 to 1%, which is what we see now. How far would that go, how fast would that go, and how many people would die. So this is here at 5 months, and if unbridled, would be at 30 million. So that's the comparison that we're looking at with this virus. Now for Europe, I think really the message hit home when northern Italy was really getting into problems. So we've had a trickle of travel-associated cases from mid-January that were actually stopped with just regular public health measures. So people were asked to report if they had signs and symptoms after travel, in that case in China. They were then put in quarantine, and through that, several of the travel introductions stopped. But a round of travel introductions in Italy went under the radar, and there was a big amplifying event, particularly in a hospital. And then there was widespread circulation in a couple of villages in northern Italy. And 23rd of February, northern Italy locked those towns down. And this was coinciding right with the winter sports vacation for the Netherlands and other parts of Europe. And obviously, what happened is that what we've learned is that the virus was not confined in those towns in northern Italy, but already also was circulating in these winter sport resorts. From where there was massive seeding across Europe, including the Netherlands. And this is what these viruses do. So you see a trickle of cases and then a super spreading event. So an opportunity where there's many people, where there is, conditions favorable for spread, for coughing, for droplets, and that's where you see the big dissemination. And we've also studied that. So you can do that by what is called genomic epidemiology. So what you do there is you look into the genetic code of these viruses. So here is a cartoon of the virus. This here is the RNA, the genetic material. It's protected by this lipid layer with some proteins on it that the virus uses to dock on cells. But if you then use this code and characterize it, sequence it, you can use that as a map to follow the dispersal of the virus. So if you do that systematically, you can start to see where did this come from, and can start to link cases and understand transmission pathways. So this is what that looked like in our country. So we've been doing that from the start. So here in the center, what you see is this sort of fuzzy bit. You cannot even see it, I think. Can you see it? A little bit. Here. Well, believe me, there's a lot of dots there. That's the original virus. And then what you see is that because that was seeded in different parts of the world, these viruses mutate when they replicate. So you start to see slightly different branches of viruses. And that tapering out is what we can use to see if we can understand how the dispersal is. So here in purple, you see virus from people in the Netherlands that had traveled to Italy, really in the first week when we had cases. And you immediately see this is not a single virus, it's not one outbreak, but it's a whole bombardment of different viruses, and that's exactly what told us this is already widespread, something is going very much under the radar, we need to do something more than what is happening now. Now that same methodology you then can use to look at outbreaks. We've heard a lot about outbreaks in hospitals, in nursing homes. Are they really outbreaks, or is that people in the community bringing viruses in? So that's the type of studies that have been ongoing. And yes, we've seen particularly in nursing homes outbreaks because for quite a while it was not possible to protect people good enough with because of shortages of protective equipment. Pretty tragic story. You can also look at this event here. So what you have heard is that we've seen the virus introduced on mink farms. Ferrets are being used as an animal model. Mink are related animals. And we still have something like 130 or so mink farms in the Netherlands. And right now, 17 of them have the infection. And the animals have been killed, culled, is what it's called. But so these animals are quite susceptible. And again, what you see here is here are the viruses from people in the vicinity. Here is a virus on one farm. Here's a virus on a second farm. And in orange, you see then viruses from people living on the farm who got the viruses back from the animals. So human to animal to human transmission. Quite flexible virus, and of course, a situation we don't want to see with an evolving virus like this. So what we have experienced is a wave, the first wave, maybe the last wave, but a wave. And here's the Netherlands. So this is so we are clearly on the downslope, fortunately, with very few cases now, new cases hospitalized each day. But I'm showing this to really emphasize that this is only in Europe, and there's big increases in circulation and in impact in big parts of the world, particularly in the Americas, all of the Americas, in Africa, and in parts of Asia. So very much an ongoing wave, and the big question will be whether this then will turn around and be back introduced into Europe once we lift our control measures. And it's also important to realize that if you look at the counts and the statistics right now, that this is as good as the amount of testing and the capacity of public health that is available. So this, we've had a lot of discussions in Europe of testing and why don't we do this more and more. This is the global picture. There's regions where there's no data whatsoever. There's regions that do report but have very little testing capacity simply because they cannot get the materials. And then there's regions like Europe and Canada where quite a bit of testing is going on, and some other regions. So that's the COVID epidemic, and we're of course still in the middle of it, but I would like to step back a little bit because it's not all coronavirus right now where we are, because these are infectious diseases, and infectious diseases happen constantly. This is a system that's called Health Map. You can look it up. And what they do is sort of web crawl, and they look for messages, newspapers, reports, websites that mention something about an infectious disease outbreak, a case, a publication, and that's collected. So, and this is the output for 1 week, and every dot is a message about infectious disease. And you can just click through them. So here, Turkey, a lot of the information is about corona, but there's also something here that warns for a tick-borne disease, an increase in a tick-borne disease, a hemorrhagic fever that is transmitted through ticks, and they warn, hey, we see more and more of these, be careful. Here, similar in Congo, yes, there are coronavirus messages in there, but here cases of plague, cases of Ebola in that same region. So I'm showing this to really bring back, like this, we are not now all focusing on corona, but there's more out there. And this is an image that was put together by the Wellcome Trust, just to say, while these kinds of infectious diseases are rare, so the number of cases are not that big, we have to be aware that they happen. So these are all smaller epidemics that happened. This is a list compiled for 6 years here with all these exotic diseases. There's cholera, Zika virus, chikungunya, Nipah, MERS, yellow fever, and what have you. And what do these diseases have in common? Well, there is, by now, there's a lot of literature. It's of course very difficult to really prove, but there is consensus in my field that we see an increase in epidemics. And for people interested, I can show some literature, but and that increase in epidemics is attributed to, well, essentially change. The global changes that we see. We see massive population growth, people moving into mega cities, particularly in parts of Asia and Africa. With that, also social disbalance and mega cities that really, where the infrastructure is not up to par to really have all these people, so you get hygiene problems. And mega cities where people need to be fed, so you get them surrounded by farms, high density farming. That farming needs to be done somewhere, animals need to be fed, so forests are burned down and things like that. So all this impact, that has an effect on the risk of transmission of infections. Viral infections are quite good at that. So wherever you see change, you have to start thinking about what that does to the ability of infections to emerge. And what we have learned from, ooh, thanks, nice. Cheers. So what we've learned from, that's a group here from the EcoHealth Alliance. They have done quite systematic look-backs of many of those disease incidents over the past decades. And their estimate is that around 70% of new infectious diseases come from the animal world. And the animal world is whatever is crawls and flies out there. particularly virus-rich are bats and rodents, not because these animals are peculiar, but because they are so diverse. There's a huge diversity of species of bats and rodents. And the thinking is that you have all this virome, the animal virome, the wildlife virome, which just is there, but from where every now and then you can get a spillover directly into people or a spillover into an animal type that is that people are more closely living with, and that can also be indirect through vectors, ticks, mosquitoes, sandflies. And the problem is that the way things are organized currently, we see these things when people are getting sick, typically. So, after a lot of things have happened in the ecosystem already. Here's an example. So that's the family member of COVID, MERS, Middle Eastern Respiratory Syndrome, another coronavirus that popped up in 2012. And here there has been a very detailed reconstruction of what happened, what led to this disease emergence. And at the root of it was a complete transition in the way dromedary camels are kept in the Arabian Peninsula. So this is the traditional style, Bedouin style. So these animals were, families would have a couple of animals, and the animals were free to roam in the desert, finding their own way, finding their own food. But in what you're looking at here is the boulevard of Doha in Qatar, and what you see is a very wealthy city, and this has been developed in a very short period of time because of oil and gas findings in the region. So we've seen explosive growth of GDP, and then what happens is that if people in the region become wealthy, then those dromedary camels, they are a deep cultural tradition. So people start to massively buy dromedary camels. So what we've seen is an explosive growth of the camel population, so much that the government banned grazing, and in no time this free roaming dromedary animal was a farmed and confined animal with big groups of animals together. Now, one of the big dissemination events here that we reconstructed was the racing business. So in this system, MERS was found, another virus, coronavirus, causing similar disease to what we see with COVID, but in this case, 30% of people that get the infection dies. So luckily, this is not so transmissible because it is a very nasty infection. And here, what we've learned is from looking at, if you then see what are all the animals in that region. So this is how that process goes. You see a new infection. You want to know where does it come from. You look at what are the animals that these people have been in touch with, and here we see animals that test positive for antibodies to this infection, and that's how the first hint was found that dromedary camels were possibly the source, and then again by looking at the virus genetics in a human and looking at the animals on the farm that he owned, the link was made that this was a virus directly coming from dromedary camels and causing this infection. So this is the system where this was going on. So this is an aerial view of a racetrack, a camel racetrack. And what you're looking at here is it's a racetrack, so there's a sand track and there's a road next to it. Because racing now, it used to be done with children as jockeys, but that's prohibited, so that's done now with robots. And the owners sit in SUVs, they race alongside, and they steer the robot with a joystick. That's essentially how it works. So that's what you're looking at. And these racetracks have a whole series of compounds right here. These are probably 1,000 little farms. And each farm has a couple of houses, has camels, has sheep, has goat, and has a deeply rooted hospitality system. So what happens here, you see international races, so camels coming in from all over the region, and they are then co-housed in these little barns with the local animals. And that's the system really where if you test the animals there during a race period, every other one of them will test positive, actively shedding this virus. It's a setup that really triggers dispersal of viruses. Here's just to show how, well, these are pets, really. People are very proud of them. These animals, if you walk onto a compound with an owner, these animals come storming just like a dog. It's quite impressive because they're quite big, but that's how they are. And also a tradition is that you then offer hot milk, which means milk directly from the camel without heating, without pasteurization. So that's a very nice way of getting your infection. And other things. So in these disease outbreaks, you always have to think what can be going on. So what we learned from this is from this guy. That's a Sudanese MD who has been studying MERS for a long time, and he did his PhD just last year with us. And he's showing the sales of urine, camel urine, because that is considered a medicine, and people drink it. And that's something that we would never have thought of, but of course then people were testing, can this be a source of the infection? Is this a way of getting this virus? So MERS we've seen also exported, just like the initial stage of COVID, mostly from this Arabian Peninsula where we see human cases to parts of the world. We have seen that MERS is present in many, virtually in every area where there is dromedary camels, but not human cases. But there have been some secondary human cases outside of the region, and there's a very big one that happened in Korea. That's what you see here. These are the notifications to the WHO. Not a whole lot, 2,500 since 2012. But these peaks here are hospital outbreaks. So yes, this is a constant trickle, a spillover, one after the other. But sometimes, if this virus is in the wrong circumstance, you can get an outbreak in hospitals so far. And in red, here's a big one, and that was in Korea, where one person triggered an outbreak with almost 200 cases. And that's because of a very cultural thing in Korea, and that's called doctor shopping. So if you are sick, you visit a couple of hospitals before you decide where you want to be treated. And this person did that while being infectious, and during that process seeded the virus into 5 hospitals. So there's been some discussion, why is Korea so good at controlling COVID? Well, I think this may in part be it. They've had SARS, and they've had this MERS outbreak not so long ago, where here they put 16,000 people into quarantine just for one outbreak. And these were images from that time, so not so long ago, 5 years ago. Last example, I think, yes, last example is of a different nature. So we see all this change, and then there's one element of change that I also want to mention, and that is climate change. So we are seeing change in, well, the temperature conditions, the climate conditions globally, and that may have an impact on infections that are transmitted by mosquitoes. So here is an example. This is already some years ago with an Asian tiger mosquito, which the name says it is Asian, and it's tropical, subtropical. But there was a period when these plants, lucky bamboo, all of a sudden became very popular, and they were imported through ships directly from southern China. And these are plants that need a lot of water, so they have little puddles of water in the armpits, if you want to call it, with mosquito eggs. So once these containers arrived, if inspectors would open them, they complained about all these mosquitoes flying out, and they were Asian tiger mosquitoes. Now, currently, our conditions are such that real establishment of this mosquito is not so likely, and there is an active program monitoring program for them, and if they are found, then they are destroyed. But in southern Europe, that process has failed, so these Asian tiger mosquitoes are now, they started to be introduced in Italy, but they are now making their way up into Europe. And as you can see here in the areas in yellow, we see repeated introductions of them with increasing frequency. And they're in the entomology world right now, I think the consensus is that this is not going to be stoppable on the long run. And that is again related to temperature and climate change. So here is a modeling study that has looked at suitability of different parts of Europe for establishment of this mosquito. This is the situation now, so where you see that most of Northern Europe would not be considered suitable, but the expectation is that that is likely to change. And with that, so if you have establishment of this mosquito, you can also have introduction and amplification of some new exotic viruses that we currently don't have. Now, that is, so we see that, and there are programs, and so you can say, okay, well, then if you see that coming, just look at it and make sure it doesn't happen. But if you then ask the question, can we predict what will happen? That actually is quite a complex question, because this is yet another animal species. It has its own lifestyle, so it needs specific habitats. Whether or not they can replicate viruses depends very much on the species. It depends on the immunity of the mosquito. It depends on what other viruses they have in their system. Trying to understand whether or not this is going to happen in Northern Europe is quite a complex area of research. But there's warning signs again. This is the Zika forest in Uganda, where in 1940-something, Zika virus was found, a mosquito-borne virus that has been there forever, was maintained in a cycle between monkeys and mosquitoes, and every now and then a person would get sick. But that virus has really come to the front page when it was introduced in Brazil in a very short period of time. And this was the previous, so we've seen that corona is called a public health emergency of international concern. This was the previous one in 2016, because then Zika all of a sudden was swarming for reasons that still are not entirely clear. And what happened is that in Brazil, we started to see a birth wave of babies with severe malformations, because if this virus infects pregnant women, it can infect the babies and lead to reduced growth of the brain, and with that small size skulls. And these babies are very severely disabled. And this is what happened. So here's where Zika used to be. but it has started to swarm, and that swarming is really along the paths of expansion of certain mosquito species until it eventually made it into South America through Brazil, where it really spread very fast because there the conditions were very favorable and there was no immunity to this virus. Now that may seem far away, but this is a related virus. It's a direct family member of Zika virus, a flavivirus, but this time a virus that does not infect people, but in fact birds, blackbirds to be precise. And what we've had is, in the past few years, a massive epidemic of infection with this virus, so a mosquito-borne outbreak in blackbirds in Northern Europe. And that is a warning sign that these outbreaks can happen even in this part of the world. So that's why, so these are some examples, but the whole, the sort of the bigger picture is that because of all the change that we see, we really need to be preparing for new disease outbreaks. And that's a situation that has led the World Health Organization to develop a list of priority diseases. This is their list, the updated list from 2018. As you can see here, MERS and SARS were on it. And this priority list is there because WHO advocates that we need to start working on preparing for these kinds of diseases because they are out there and are likely to expand in the future. But on that list is also disease X, and that's an alert to say these are the ones we know, but there may be others out there, and we really should be thinking about generic preparedness. Now then, of course, that's a very tricky question, so how do you do that? And the focus of the work that we are doing is, can we really, by understanding what's going on in the animal world, in wildlife, and the impact on that, can we maybe start to predict where risks of outbreaks is increasing, because that then would be places where you really could start targeting your surveillance, for instance. This is also a call by Anthony Fauci. You can see him nowadays on TV looking very unhappy because he has to brief Trump, but Trump doesn't take much of advice. But he has been advocating to also start exploring the world of artificial intelligence and the new tools, the catch-all tools, the metagenomics, the different technological developments that really could be used to get this increased preparedness. I'm particularly a fan of doing metagenomics, and what is that? That is, so you take a sample and there's a certain technique where you can characterize all the genetic material that's in that sample. So that's all the viruses, all the bacteria, all the parasites that are in there, and then use that as new information. And if you start doing that, then from those types of studies, what we're learning is that what we know is very little compared to what really is out there. So those are techniques that are increasingly being explored for preparedness research. And then, of course, if you would have those kinds of techniques, combine that with data types that help you understand where is change happening, where is risk increasing, when do we need to raise a flag, and start looking deeper. So that's, I'm coming to an end. So that's the way I think we should start looking at the world, so that realizing that the health of humans, animals, and the environment that we live in is interconnected. That's this Gordian knot. We share that with the pathogens here. That's a little piece of DNA. And whenever you change something, you put these systems under pressure, then that will affect the circulation of pathogens in them and in all these systems. So that's why in different types of projects that we have, we are trying to study this really combined in the Netherlands Center for One Health. So for now, regardless of what petitions or not, please keep your distance for now. Because I do think, going back to Corona, that this virus is still circulating in big parts of the world, so we should not get too lax about it. Then look at the future with optimism, I would say.
Thank you. Thank you very much. Thank you very much, Marion, for being here in the middle of this hectic time. Thank you also here for being here, and thank you the audience at home. My name is Job de Vries. I'm a science journalist, been covering this topic also extensively for almost since the beginning. Time for questions. If you have any question here, don't hesitate and walk to the microphone. And you can stand there with 1.5 meters in between and waiting. Of course, all the questions from you and from the people at home who can send in through WhatsApp, the numbers in your screen. Those are like the most important ones, and we also have some questions. There's already one, so please go ahead.
Yeah, thank you. Could you tell us a little bit about the rate of mutation of this virus? In other words, point mutation, block mutation, and also the implications for the potential development of a vaccine?
So right now what we see is a fairly stable virus. We see mutations, but not a whole lot. So we see about 1 mutation in 2 weeks, which is quite limited, and no mutations yet that you would worry about in case of that could translate into different properties. Right now, I don't think we have to worry about vaccine, but if and when a vaccine is introduced, yes, and this virus keeps circulating, we will have to start looking for updating to see if this virus escapes from immunity, which my prediction would be that it would do. And the reason for that is that it is a respiratory virus, and we know if you look, for instance, for influenza, that people do develop immunity, and you can induce some immunity with vaccination, but it's not sterilizing because mucosal immunity is difficult. So you still get infection, and that means you have a selection force on the virus. And what that does is favor viruses that have mutations that can escape from that condition. So that's a sure way of getting drift mutants. So if we see either natural immunity develop in regions where the virus has circulated a lot, or vaccine immunity, we really have to start monitoring for those kinds of changes.
Maybe in addition to that, you mentioned that that people, after they will have had that vaccine that might be there in a while, they could still develop the infection, possibly. Would that also mean they would still be able to spread it? Because that's also a thing that we've been discussing a lot.
Yes, that's a big question. So it's hard to know at this point. There was a first vaccine challenge study in monkeys, in macaques, with the runner-up vaccine that is now being tested in people. And what they did find is protection from disease, from pneumonia, but not from shedding. So that's not so good news. You would ideally have something combined. On the other hand, what we see in patients, if people may be shedding the virus, but if they are also antibody positive, that virus cannot be cultured anymore. So there may be still an effect from vaccine on shedding. But those are important questions. And
yeah. And you would ideally want to vaccinate the vulnerable people and not protect them by vaccinating others then.
yeah, exactly.
If you have any other questions, don't hesitate. We are already having a couple of questions came in during the talk. So this one is quite related. It's sent in by Willem. So do you think that this virus, SARS-CoV-2, will become more or less harmful or virulent over time? Will it become more like the other ones, the other coronaviruses, or will it still be the exception?
Well, the expectation would be that it becomes less harmful, but you never know that for sure. There is some experiments, so we do see, you saw those clouds of viruses, so people have compared the two, and in the lab it looks like one is maybe producing more offspring than the other, so that could be a bigger spreader. But there's no evidence of that really in real life yet.
Are there any examples in your lab in Rotterdam that are trying to go after that?
Well, we are of course looking at what happens. So if you see these, so we see different groups, clusters of viruses. So we try to grow them and see, do they behave all the same? Because we don't want to. Yeah, we don't know. There's not so much information. One aspect is that we know that these viruses require a certain receptor, the ACE receptor, you know this. But it looks like there's a second mechanism involved. And some viruses use this one preferentially, some seem to use the other one preferentially. So there is something ongoing, but we're not sure what that means.
Okay. I see one here.
Hi. Yes, I was wondering that given that the lifting of the restrictions is primarily an economic decision now in the coming months, I was wondering what your opinion as a virologist and a scientist is on that, and whether you actually do support that with science, and given that a second wave is appearing in quite a lot of the world, what would make the Netherlands sort of an exception to this?
So I think a lot of the world is still in the first phase, that's for sure. Yes, I do support lifting of measures, but carefully. So, because what we do see is that the number of new cases really has gone down, and has stably been going down now for a while, to the extent where, people that test positive, new people that test positive, they are contacted to see who they've been in touch with, and that system seems to work quite well. As long as that is maintained, then I'm comfortable with more movement and more lifting. But we really need that, and we also really need that people get tested if they feel sick. And that's where I do worry a little bit, because I do hear that maybe that is not so popular. The testing is not very nice. But that's two arms of the system. So if people do that and you know, follow the recommendations, maintain the physical distancing, get tested when they're sick, I think we can do this safely, and all of that is in place to understand that as soon as the virus is picking up again, that we have to do something.
Can I follow up?
Yeah, sure.
Yes. And what would you expect, do if that virus does start to pick up again? Would the measures then be extra, severe with regards to these measures?
It depends a lot on where it happens and how it happens. So, for instance, in the thinking about what can you do, so initially there was like a cap on 30 people in a room like this. It does not really make sense. It makes sense to say, keep the physical distancing, keep the hand hygiene, but do it so that you can do that and then fill the room with as many people as you can do while keeping this. If then an outbreak happens, you know who have been here, because that's part of the situation that you want people to register. So then that control would be just in a defined group of people and their friends and their contacts. So that's how it is going to work if that whole system works well.
I see it next one coming. I have one in between, maybe a follow-up on this one, because you already mentioned, I hear that people seem reluctant to test, and you probably hear other rumors. So how do you try to keep in touch with all those rumors and questions and things in society without sort of getting soaked up, because you could probably be debating and reading and hearing stories 24/7.
So it's hard to know for sure, of course. So what we do see is a lot of people do get tested. What we have seen is a lot of, lately, a lot of children got tested, and that had to do with children often have runny noses, and they could not go to daycare when they had runny noses, so people wanted them tested. There is, so RIVM does polls with a substantial group of people to hear about, would you test yourself? It's anonymized. Would you get yourself tested? What if you have a friend who tests positive? Would you do that? So those are the kinds of systems that we have to look at.
Mm-hmm. Okay. Here's one question.
What's in general the idea when a pandemic or an epidemic evolves? Does it naturally go away, or is there a line in that? Or is it always a different type of evolution?
So pandemic literally means all over the world, so my expectation with this virus is that we won't get rid of it simply because it's so widespread. So and then the most likely scenario is that after a couple of bumps, that becomes number 5 of the winter coronaviruses. That's I think the most likely scenario. Do I know for sure? No. If you look at so we've had a couple of flu pandemics that have been studied quite well. And if you look also at the pattern of epidemic waves, they have been very different with every pandemic. So, and this is a big uncertainty. What we need to understand is how much, so if people have the infection, do they develop immunity? We know that now, but how long does that last? And is there some type of cross-immunity? For instance, is that maybe a reason why children don't often get sick? They get infected, but they far less often get sick. And those factors, if you know them, then that determines a lot of how you can predict how this virus moves forward.
Another additional question there, because you mentioned a lot of people will develop antibodies after they get immune, but there's also a large group that don't seem to develop symptoms. They might not develop immunity, but they might also not even need it because for them it's so mild that they didn't even experience it. How do you see that in the bigger picture of those waves and of the spread of the virus?
So that's the pyramid question, right? So how many people are below here that have hardly any symptoms or no symptoms? Yeah. That we don't know yet. So far, it looks like also people so you can learn that from household studies, for instance. So where you test everyone, also the people that didn't get sick, and you do see that there's more people with antibodies than people that got sick. Is there a category that doesn't even get an immune response? There's now a paper out that suggests that, but I'm not convinced yet.
Yeah.
So I think, like with any infection, you have a group that has asymptomatic infection. I would not be sure to say that, oh, once asymptomatic, always asymptomatic, because that's typically not how it works.
Yeah. Okay. And I have a next question from online. What's your advice for young virologists or medical scientists in these times?
Study One Health.
And One Health is?
So it's really an interdisciplinary science that starts to think, okay, how do these things interact, and what do we need to come up with to study these things in connection, rather than human health, animal health, that move in parallel worlds.
And what would be a very good question to study for a PhD student or someone who starts?
Oh, there's an incredible amount of... So we just started a project with 26 PhDs in 9 universities that all... it's mandatory to have a second supervisor in another institute because we want this cross-fertilization.
Yeah.
And if you, it's incredible what you see happening if you do that. So you see all these ideas that I think about human health and virology, and I sit with a person who is specialized in mosquitoes and flies feeding on dung, and then you sit for a half an hour and you have all these ideas. That's the type of environment that I would look for.
Okay. Something very different. A question from Edwin is more on the policy. How, on a scale of 0 to 10, how successful would you consider the Dutch response to the COVID-19?
Well, comparing globally, I don't think it went too bad. If you start from a position this should not have happened, of course it's a 0 because it happened. What we've seen is, of course, that there are gaps in our preparedness, big gaps in our preparedness. That's clear. So I would say 6 minus.
So could have been better. And
could have been better.
What gaps do we need to fill for the next?
So
well, maybe the next stage, but maybe also the next pandemic. Possible
so
pandemic.
One thing. So so I've been involved in drafting pandemic plans for flu, and then so a lot of what happens is not so surprising. What to me was something that I did not realize is market failure and how that can shut things down. So we really depend
with supplies
on supplies.
Face masks, tests,
exactly.
all those things.
Yeah. So that part of preparedness I think is important, but also what you see is I think the public health people are more prepared than clinicians. So hospitals, and for them this was what's going on, and
public health was also quite underfunded, right? Recently, like the GGD.
Yes, yeah. So and this has been a problem. I mean, the blueprint list of WHO was developed after Ebola, saying, okay, we must invest in disease preparedness because we can expect these things, but it's not been popular to invest in things that may or may not happen.
Yeah. Then maybe you already mentioned WHO. There's also a debate on how WHO has been performing, the World Health Organization, and what their role should be. Can you comment on that? How do they seem to perform, and how in this current, well, political age?
I think the work at WHO is hugely complex. So they cannot walk into a country and say, hey, I'm going to tell you what to do.
They have no teeth.
No. They are completely dependent financially from contributions of the countries. And those contributions sometimes come with a tie. Look at what happened with the American contribution. So given those circumstances, I think WHO is doing an outstanding job, actually, because we cannot expect them to really control these things, because what they can do is signal, they can find information, they can pull expertise together, but they cannot go in and control the situation. That's a national responsibility.
Do you think they should get that power or that responsibility? And can it?
Well, it's hard to know. I think, so that means giving up national sovereignty, right? We've had that situation when we had a Q fever outbreak. There was big criticism internationally. Hey, what are you doing, Netherlands? Are you enough on top of this? Would you want an international organization to come in and say, out of the way, we're going to do this?
Yeah.
I don't know.
Difficult debate. There's a question here, and we're running out of time, so we'll try to squeeze in. Go ahead.
Yes, the last few days to weeks, we see a lot of new cases in Houston and, for example, Florida, which is going pretty rapidly. What would you advise Trump to do?
Well, he doesn't take any advice, so I think they really need to go into serious control in I think it's 21 states. If I hear also what's going on in Houston, I wouldn't be surprised if they get into the code black scenario that we've been discussing in some of these states. So, yeah, I think they really need to go into very severe control measures.
Because if they don't, what do you expect to happen?
Well, the overflowing healthcare system. So obviously the healthcare system in the US has more bandwidth, but what I hear is that in several of the states that's filling up fast. So they're already opening up pediatric wards for COVID patients. And well, we know from what we've seen in the Netherlands is that, you know, there's not a long time between the first 10 and the filled up hospitals. So I think they really are in trouble.
Thank you.
All right, I think there's more questions. I think we might be able to arrange to answer these questions through WhatsApp. Maybe we could arrange that because the taxi is where your taxi is about to come. So thanks again very much for being here tonight. Thank you all. Thanks for watching, and also thanks Paradiso for organizing this. This live stream will be online until Monday, so you can be watching it back. Other people can watch it back. And hopefully we'll see you again at the next, maybe normal Science and Cocktails next time. Thank you very much.